My name is Xiang Zhang. I am a PhD student in computer science at Professor Chad Meyer's lab at the University of Minnesota. I have collaborated with the research teams at University of Toronto and University of Michigan for the project I'm presenting here, a machine learning approach to identify novel antifungal targets in Candida albicans. Candida albicans is an opportunistic fungal pathogenic yeast that can sometimes lead to deadly infections in humans. Therefore, understanding which genes are essential for growth of this organism would provide opportunities for developing more effective therapeutics. However, unlike the model yeast, it is much more laborious to construct mutants in Candida albicans. In addition, according to the Candida genome database, the Candida albicans genome should consist of around 6,600 genes, but there is very few official information about the essentiality of these genes. Accordingly, in order to generate a comprehensive assessment of genes with evidence for essentiality in Candida albicans, we proposed a machine learning pipeline based on a set of functional genomic features and a gold training standard called Grace Mutant Library. More specifically, we have collected and engineered features from different sources, such as gene expression datasets, sequence features, including codon adaptation index, S-seroVCA data, as well as 6 features from a recent transposon mutagenesis study in a stable haploid genetic background. In the Grace Mutant Library, each strain carries a precise replacement of one of the 2 alleles of a gene with an auxotrophic marker, while the remaining allele is under the control of a tetracycline repressible promoter such that the gene expression can be repressed by the tetracycline analog doxycycline. Based on this training standard, we determined the essentiality of 2,327 genes, and about 500 of them were experimentally confirmed as essential. These serve as the input to train, optimize, and test a random forest model, which finally outputs the prediction scores for all the genes we have on hand. Based on the predictions, we prioritize the construction of an additional set of 866 strains, which we call the Grace V2 set. Our strategy is to train and optimize the random forest model on 80% of the genes from the Grace library, and the statistics on the right summarize the test performance on the remaining 20%. With a stringent cutoff of 0.5 for prediction scores, we estimated an average precision of 0.77 and an AUC of 0.92. After that, we retrained our random forest model on the whole Grace gene set using the optimal hyperparameter setting, and got the permutation feature importance for every single feature. Finally, we predicted the essentiality for 6,638 genes. To assess the accuracy of our prediction model, we selected 866 genes for validation, covering the range of the prediction scores. We discovered essential genes at a rate of about 64% among these new predictions relative to an expected background rate of essentiality of about 20%. We can also see that our predictions are able to discriminate the essential genes based on the distribution density of the prediction scores for the validation candidates. We also have the summary statistics for the predictions from our optimal random forest model on the Grace V2 validation set. We achieved an average precision of 0.66 and an AUC of 0.95. In summary, we built a generalizable machine learning model to predict gene essentiality of the majority of Candida albicans genome. Given the strong results, we helped facilitate the experimental validation of new essential genes. In total, our group has confirmed 621 essential genes in Candida albicans, and among them, we identified several essential genes that lack human orthologs and thus represent novel antifungal drug targets. That's all for my presentation. Thank you.